Good afternoon, ladies and gentlemen, and a very warm welcome to this session on ethnobotany on this Wednesday, the 19th of January, the third day of the conference. I must say I'm very impressed and enthralled and excited by how well we're represented by ethnobotany in this conference. And in this session, we have 4 speakers presenting. Unfortunately, Palesa Mokoena is unable to present her talk, which was the second planned one in this session, but we have the 4 other talks which look to be very interesting. We will start the session with Mkhlatse Mogale from the University of Johannesburg, who's going to be speaking to us on an inventory and analysis of medicinal plants used by the Bappedi in Limpopo Province, South Africa. Mkhlatse, the floor is yours.
Good afternoon, everybody. Let me quickly share my slides. Are my slides visible?
You can go ahead.
Everybody. Presenting, my name is Mkhlatse Mogale, and I'm a UJ student. I'm doing my PhD supervised by Prof. Ben Eric van Wyk. And today I'll just be presenting on a small part of my PhD study, and it's on an inventory and analysis of medicinal plants used by the Bappedi in the Limpopo province. So just a little background on Bappedi. I just want to show you some of the artifacts from the Bappedi culture. In your turf of you see a grinding stone, which has been used for many, many years, as you can see from the hole that has been created in the stone to grind some medicine by a traditional healer. And to your left at the top, you have a sieve, which they use to make traditional beer, and they store them in those 2 containers. And then you have your traditional attire from for a woman that has been initiated. And at the bottom, you see on your left, bottom left, you have the traditional itinerary from from one of the chiefs in the Sekhukhune area. And on your bottom right, you have a traditional herbalist, actually, who is showing me how to prepare one of the medicines he's going to administer to one of his patients. So the aim of this talk is it's a literature review of medicinal plants used by the Bappedi, which will be enriched by my own data. And the objectives will be to see how many medicinal plant species have been recorded to be used by the Bappedi, and also medicinal plant use records have been recorded for the Bappedi, and also to see how many vernacular names have been recorded for the Bappedi medicinal plant species thus far. So Bappedi are predominantly occurring in the Limpopo province. The province has 5 districts, namely Capricorn, Waterberg, Sekhukhune, Vhembe, and Mopani districts. And Bappedi predominantly occupy 3 districts, which is the Capricorn district, the Waterberg district, and the Sekhukhune district. The Mopani district is predominantly occupied by Tsonga, and the Vhembe district is predominantly occupied by the Venda. A little history. Originally, Bappedi were part of the Tswana-speaking tribe, which was located in the northwest. The name of the tribe was called Bakhatla, and due to internal disputes, a group of the Tswana decided to split away from the original group, which ideally became known as the Bappedi, and they moved far the area today known as. This happened back in the 1600s, but the area was formerly named Sekhukhune in the late 1800s. And one interesting fact is that to know is that on their travels, they came across a porcupine. And if you know, the Botswana totem is a monkey, which is called a khabu traditionally. And so Bappedi decided to formally detach themselves from the Tswana and took on the noko, which is a porcupine, as their formal totem. So I'll just show you some of the landscapes that I came across from some of the localities I've visited in the Limpopo province. So the first area is called, the first village actually, it's called Muhelere. which is located just past Liwahomu in the Limpopo province. And this is a picture I took while I was on a walk with one of the residents there. He was showing me the area around. And we came across an interesting plant called Lihoromecho, traditionally, and scientifically is known as a Pilonas. This is used to treat respiratory conditions, and they would crush the leaves and wrap them up and smoke them to treat some of the ailments they have for the respiratory. And also it's a very popular male aphrodisiac. This is a village in Sikukune named Pokwane, and this is the landscape that you can actually see from when you walk out just behind the village. Unfortunately, the area is by one of the by a Lopholinena corifolia plant, and it's actually causing problems in the area. But then I managed to see a couple of interesting plants. The first one on your bottom left is called Mushwani, sorry. And it has red roots. So the red roots, they signify that, traditionally they signify that the plant can be used as a traditional medicine. And the traditional healers actually use the roots in a number of combinations to make medication for, including blood-related diseases. And the plant you see in the middle there is called Tlhabo Magotwana. Scientifically, it's called Neclidium zeheri. And it's a very strong plant that they use for skin infections. They would burn the plant and the ashes, they will rub the ashes on the skin. And to your far right, you have a plant called Matlapaneng, scientifically known as Perinari capensis. And nice thing to know about this plant is that the name actually explains where the plant grows. The name literally grows in English, and it's used for blood-related ailments like high blood pressure. And this is a village I also visited. It's called Senwa Barwana, which is a new name. In the old name, it was called Bochum. And the people there took me next to this hill here, which is traditionally significant because they believe it has the ancestors usually walk around there. And they normally say you can't go to the hill because that's when it's 12 midday, nobody goes to the hill, and you go there just after 12 from 1 o'clock onwards. And around this hill, we found Cassia abbreviata, which is popular in the area for treating measles and asthma. So I also looked into a lot of literature on in connection with the ethnobotany and medicinal plant uses of Bappedi, and I came across a lot of published articles by Dr. Semenya Zibua, who just passed away recently, and who published a lot of work on the medicinal uses of Bappedi and was working with a lot of his colleagues. One of them is the gentleman there is Dr. Potgieter, who's a professor at the University of Limpopo. And I also published recently for my master's, the ethnobotany of Central Sekhukhune land, which I also incorporated into this inventory. So all this data I put in a table you can see on this slide. So the table has several columns. The first column we find is the species. Second is the plant parts used from the plant species. And then you have the medicinal uses from the Bappedi. And then the last column is the medicinal uses by other cultures. So for example, the first plant there is Artemisia afra, which comes from the Asteraceae. Locally is known as Lengana or Milanchi. And the leaves and roots are plant parts which are used. And you can see some of the annotated plant juices. The leaves are boiled and the decoction is drank to treat to treat TB. The roots are used to treat impotence, and there are also other uses like treating asthma and malaria, if you can read there. And also, the plant is similarly used by isiZulu culture. The leaves are actually used to treat respiratory disorders and also to treat loss of appetite by the Zulu. The way I compiled my table, which gave me a total of 286 medicinal plant species which are known to be used by Bappedi. So when I was further looking into the table, I found that the species belong to 81 families. If you can see there on your left, the table on your left, the species diversity of Bappedi medicinal plant families. Most of the families belong to the Fabaceae, which you can see here is there's 28 species belong to the Fabaceae, and the second highest group is the Asteraceae. So when comparing them with other cultures, when comparing it with other medicinal uses in Africa, I referred to a paper published by Prof. Ben-Eric van Wyk in 2020, which looked into the African medical system, the whole of Africa. And if you can see here, my table and the African table agree on the number of plant species used in the Fabaceae group, which there's a large group used in Africa under Fabaceae, but group in Africa is Rubiaceae, and the third, unlike the Bappedi, is Asteraceae. The Bappedi came in as second, and fourth, like Bappedi, was Malvaceae, and fifth was the Euphorbiaceae. When in the Bappedi, the Euphorbiaceae came in as the third highest group of plant species used. Further looking into the genera of the species used by the Bappedi, most of the genera come from the Aloe, comes from the Euphorbia, the third comes from the Grevia. I looked into other cultures of the most popular species. There was a paper published recently by Mhlongo and van Wyk in 2019 on the Amandawe village in KwaZulu-Natal, and the most popular genera was from the Solanum. The second was Amaranthus and Commiphora, which is totally different from the Bappedi popular plant genera. And then I looked into work published by in 2019, which was actually on the useful plants, not focusing on a larger group of plant uses. But this genera actually agreed more with the Zulu than with the Bappedi. You can see that Amaranthus and Solanum are the top two most popular genera of species used. So I further looked into the medicinal plant species recorded from the different cultures in Southern Africa, and noted it looks like they've been very undersampled. It seems like there's a lot of work that can be done because there's only 286 medicinal plant species recorded to be used for medicinal purposes, as compared to the published book by Hutchins in 1996 on the Zulu traditional medicine, where he recorded 1,032 medicinal plant species that are used by Zulu. And Mutiti published a review of Basotho traditional medicine, where she recorded 437 plants which have medicinal uses to Basotho. In ethnobotanical research, published that there are 384 plants which have medicinal uses to Vavenda. And Harley and Van Wyk published 287 plants which are used for capable medicines, which is in the Little Karoo. And you can, if you can see here, like Bappedi are really, really undersampled, and I think there's a lot of more to be discovered as the journey continues. I also looked into the plant parts which are used by Bappedi, and there's a lot of medicines that are used from the roots, combinations from the roots, and the second highest is the leaves, and the whole plant comes as the third highest used plant parts. Something interesting is because that they, when I was talking to some of the people during my survey, I found that they prefer the roots because the roots are plant parts that are regularly available throughout the year. They have a seasonal significance because the Limpopo is a relatively dry area. It's a savanna. So leaves are not regularly on most of the plants. So the roots are the preferred plant part to be used for medicinal purposes, as it's available throughout the year. And the least used plant part are the seeds, with the fruits as the second least used plant part. And a few medications have unspecified plant parts used. So there was a rough grouping of the ailments that I did. And if you can look here, the respiratory ailments that are related to respiratory ailments, the one number of medicines that are used to treat a number of those ailments. And then the second highest is the blood-related ailments. Thirdly comes the gastrointestinal and the skin. But then I had to further think about the grouping. So I looked into an article published by SAAB in 2015, which is about the classification of diseases, which is a classification system that was also used in one of the papers I used as part of my reference, which was by Mhlongwe and Van Wyk.
Apologies to interrupt. Can you shorten up the presentation? You're going a bit over time. Thanks.
Oh, sorry. Okay. Oh, so in summary, the paper publishes that there are 3 classification systems: the International Classification of Diseases, Economic Botany, and the International Classification. The reason why I chose the third one, the International Classification of Primary Care, is because this classification system allows plant uses across cultures, which the other 2 do not. If you look here, the International Classification of Primary Care actually has 17 categories which are used to classify ailments, and the 18th one is just an unspecified ailments. So I use this to compare my inventory with the Zulu traditional medicine and Cape herbal medicines. If you can see here on the first graph, you can see when you compare it with the Cape herbal medicines, Bappedi and Cape herbal medicines actually treat the ailments. But then Bappedi treatments are for stomach pesticides, but then Cape herbal medicines do not treat those. And if you look into the Zulu, the most treatments come from tonics, and the least actually, they do not have any stomach. The least is the stomach ailments. So in conclusion, there's currently 286 recorded Bappedi medicinal plant species which belong to 81 families, which the most families is the families. To 51 genera. The yellow and euphorbia have the most with 7 species. And there are 6 new medicinal plant species which I came across in the preliminary survey, and 512 documented vernacular names were picked up. And currently there are 517 medicinal plant use records. And just from preliminary survey, I picked up 27 new medicinal plant use records. And just an interesting conclusion from an article I published in 2019 that there are 170 medicinal plant use records. And in conclusion, thank you.
Wonderful. Thank you very much for a great presentation, very, very clearly presented. Interesting that going back to an area does show some use in finding new medicinal plant uses and new medicinal plants to be used. I just have a quick question because I don't see any other questions in the chat about the use of the roots. Do they ever have problems with sustainability of harvesting plant material? Do they mention that?
They prefer, as a technique that they use when they do not harvest all the roots. They prefer to harvest on one side, and then they won't cover the uprooted part. They say if you cover that side, it kills the medication. So they prefer to harvest one side but not the whole roots. So just a small portion to allow the tree or plant to give it a chance to survive.
Great. Yeah. Okay. Well, thank you very much. We can just keep an eye on the chat if there are any further questions later. But thank you again for your interesting presentation, and good luck with the future of the work.
Thank you.
All right, thanks.
Thank you.
Then our next presenter. I think that's going to be fine. All right, the next presenter, as Palesa won't be presenting today, would be Martin Mushamba, who's going to be talking about induced antibiotic resistance and staphyloxanthin as a prospective target for treatment against pathogenic antibiotic resistant staphylococci from the University of Pretoria. Welcome, Martin, and go ahead with your presentation. Are you able to share your screen?
Hi, Lindy. It seems like Martin has lost connection. So if we could possibly go to the next talk and come back.
Perfect. Okay, thanks, Jackie. Okay, so we can move on then to Tsepiso and Ndlovu. who is from Northwest University and University of Mpumalanga, dual affiliations there. So he's going to be talking about an inventory of medicinal plants used to treat and manage childhood diseases and well-being in the Northwest Province. Thank you very much, Tsepiso, and you're welcome to share your slides. Is Tsepiso online? Can you hear us? If you can share your camera and microphone, then you can share your slides. Okay, while we're waiting for Martin or Tsepiso to join us, we do have a question for Mkhlatse from Service Alberts. Thank you for the talk, very interesting. Are there any literature available where the smoke extracts of the plants used for smoke healing have been investigated? So, Mkhlatse, if you can hear us, you can put on your mic.
Hello.
Hi, thanks so much. So, is there available about smoke?
So far, I haven't come across anything on smoke extracts for any of the plants that I've come across. Maybe if they find something, they can let me know, but so far, I haven't found anything.
Okay, interesting. Thanks very much. Okay, and I see Tsepiso has joined us. Thanks very much, Tsepiso. You're welcome to share your slides and begin your talk. Remember, you have 15 minutes for talking and 5 minutes for questions. And we are a little bit late because of the last session running on. So we're trying to make up a bit of time here.
Yes, thank you very
Thanks,
much.
Tsepiso. Looking forward to your talk.
Thank you very much, Prof. I'll just request for the technical to assist me with the slide as we discussed previously. Thank you very much. This is a PhD study on medicinal plants that are used to treat and manage childhood diseases in the Northwest Province. Next. Okay. Globally, we all know that medicinal plants have played a critical role in human disease and also in ethno-veterinary medicine. Medicinal plants remain one of the indispensable resources that we have on Earth, and it continues to do so. Subsequently, so we also have childhood diseases that are also increasing in daily basis for children under the age of 5. In 2000, it was estimated that around 12 million children under the age of 12 in developing countries are succumbing to death, which is almost contributing to 70% of death in local communities or developing countries. Next. Okay, so in last year, we did a review whereby we wanted to understand how many medicinal plants that are used in South Africa. To the study that we conducted, we found that there were almost 194 medicinal plants that are used to treat and manage childhood diseases in South Africa, whereby most of the medicinal plants have been documented in the KwaZulu-Natal area. While in the study that was conducted in Northwest, we found that only the study by Dr. Asong, had only recorded 3 medicinal plants, which was not the main focus of the study, despite that medicinal plants of children, it's being utilized in that particular area. Next. So we all know that children are the future of the world, and if we don't take care of our children, it's like we don't take care of the future. So in terms of Northwest is one of the predominant rural area province with a high shortage of medicine and other conventional health practitioners, and it's also a province with large of unemployment in the country, whereby there is also a high rate of diseases according to Statistics SA. Now, in terms of the several studies that I've concluded, it was mentioned that medicinal plants, the limited studies of medicinal plants that are used to treat childhood diseases, and as we know that medicinal plants are the vehicle of living in South Africa. So the study was aimed at looking at medicinal plants that treat and manage childhood diseases. Next. Okay, so the study was aimed at documenting traditional knowledge and medicinal plants that use and manage childhood diseases in the Northwest, with the specific objectives being one, to identify medicinal plants, to generate inventory, and also to document the indigenous beliefs associated to medicinal plants. And with these two related research questions that are bound to be answered. Next. Okay, so the study is underpinned by an interpretism paradigm whereby we believed that a socially reality is constructed with the participant, whereby a researcher and the participant engage in generating the knowledge, meaning that the researcher and the participants are co-generators of the knowledge. We used the method, the mixed method, by Creswell 2017, and the study is a descriptive survey study that we embarked on with the sampling technique of traditional healers that were selected and the herbal vendors, given that they are the people who normally understand the role of medicinal plants within the community. And also we used the sampling approach, the two sampling techniques, the probability and non-probability, whereby we selected the villages randomly, but then also equally within the district that we choose. And also the participant, we choose them from referral point of view. And also prior to the collection, we actually piloted our study in Madibeng municipality. Next, with 15 participants. So the study was conducted in two districts, which is Bojanala and Ngakamudere Molema in the Northwest province. The area is almost 94% of Black people are dominant in the particular area, and its economic activity ranges from mining and tourism, with the rainfall that is highlighted there. Next. Okay. So in terms of ethical considerations, the letter prior conducting the study, we went to meet traditional councils, Barolombo Ratsidi, to ask for permission. Then we also went for the Ngaka Association, because of that, the association, that the body of the traditional health practitioners in the province. Then we applied for ethics with the HREC of the Northwest, and also the permit to collect plants with the Department of Economic and Development. And also we had a consent form which was translated in Setswana, which also articulated the following aspects that are there for confidentiality, anonymity, and principle of respect that was applied during the research, or that we are going to apply. And also because of this study was also between the COVID-19, also the guidelines for COVID-19 were also observed during our visit. Next. Okay, so in terms of our ethnobotanical, 10 villages across the districts where we had 101 participants across the two districts, using the semi-structured interview face-to-face, using the local language, which is Setswana, and also the plants that we collected, we sent them for identification in Zambi, and we used the various ethnobotanical indices such as cultural importance index, and also use value and fidelity level to determine the knowledge and also the uses of the plants that are used in the area. Next. Okay, so this is us in the field, whereby the first is when we went to the second traditional council. We were just ice breaking before we could further engage our participants. We were briefing them about the importance of saving of documenting the medicinal plants that is in the study, or that we are about to pursue. So this is the pictures of us in the field. Next. Okay, so in terms of the medicinal plants, we had 81 medicinal plants that were mentioned to be treating and managing childhood diseases in the area. However, after we sent the plants for identification, we only found that only 61 plants are the only correct plants that are being used. Hence, we have 61 plants that are used by 33 families to heal or to treat or to manage 29 different childhood diseases within the area, whereby we have plants such as, next, there's a picture that I will, yes, whereby we have plants such as Atumsomnia elongonium, which is one of the highly used plants in the area. It had a highest use value, cultural importance index that was high, and fertility level. And this shows you that this plant is highly being used for children to treat different types of ailments, starting from Sanking for Nutella, whining, and other related diseases, even diarrhea for children in the Northwest. And the other one was Euphorbia protesta, which is also the second plant that had the highest UV in the study area. And also people, participants often refer to it in terms of preference. And the other plant was this blue band phytocensus, whereby most of the participants also mentioned it. However, it was only mentioned as being important in terms of cultural value. Next. So in terms of the plant families that were represented, most of the plant families were represented were Leguminosea and Asteraceae, with 7 plants per each species. This is also found in different studies that Asteraceae and also Leguminosea are highly consist of number of medicinal plants that are used for different diseases, also in ethnoveterinary. Even the previous speaker just mentioned that Leguminosea is among one of the highest plant families that has been mentioned in terms of plant families. Next. So in terms of the life form, most of the herbs most of the plants that were identified were herbs in terms of the participants, whereby it's almost 60% of the plants were herbs. It was observed, and also meaning that the study usually deals with children. So in terms of the toxicity level of medicinal plants, it was of importance that children also have a vulnerable immune system in terms of drinking highly intoxicated medication. So also in terms of the plants that were used, they were using the whole plant leaves also, and also the under root of the plant was also highly dominant, which is also resonate with other studies that are conducted throughout. Next. So in terms of plant preparation and administration, you find that most of the plants, they used maceration, and also decoction was highly. And also in terms of oral, in terms of administration, most of the plants for childhood diseases were orally taken, which was highly also in several studies that have been conducted across South Africa. Next. Next, okay. So in terms of the conservational status, what we did after we've identified our medicinal plants, and we also did we did check them on plant list and world flora. We also went to SAAB to check whether there were plants that were red listed, or the status of the plants. In the study, we found that almost 79% of the plants still have list concern in terms of the SAAB red list, which this resonate with the previous review that we did that stated that almost 89% of the medicinal plants that are used for childhood diseases are still at the safe. However, there were 2 plants which are also listed, your wild ginger and your paperback, which are among the plants that were being listed. However, they are facing extinction, which we need to take care of these plants also. Next. So in terms of the condition of the study documented 61 plant species from 23 families that were used to treat 29 different diseases or diseases related to children. With this, it shows that Northwest consists of many knowledge which needs to be explored, and also the diversity of knowledge within the community is still vast. And also for further studies, we recommend that the more documentation should be done in the area, especially given the large area that it has. I still believe that the 61 plants have not yet reached the pinnacle of the study, and further phytochemical profiling and safety of the medicinal plant should be also dealt with in the future studies that needs to happen. Next. So I thank you. I'd like to thank the Northwest University for permitting me do my PhD with them, and also my employer, University of Mpumalanga, the Ngaka Association for giving me or for letting me pursue my studies with them, and also the SAAB for giving me this opportunity to present, the Northwest Provincial Government also for permitting me the permit, and SAMBI for identification. Thank you very much. Oh, the NRF.
Very important to thank the funders. Thank you so much for a very interesting talk as well. Are there any questions for Tsepiso on the childhood diseases? I was wondering if there are any particular plants that stood out as being used for only childhood disease treatments. Is there anything that came through there? Because as we know, so children are not small adults, and it seems that traditional healers do distinguish between treating the children and the adults, which we
yes, actually there are many plants that, because of the study, it's only dealing with children. So we were only looking for medicinal plants that were used for children. So hence we went for highly people who understand children or who are working with children through referral. So most of the plants that have been identified are only for children.
Okay, no, that's very interesting. Okay, thank you. Any other questions for Tsepiso? All right, well, thank you very much. I think in the interest of time, we can move on. Is there another speaker who is ready to present, either Martin or Chika?
I'm ready to present.
Oh, hello, Martin. Thank you. I can see your screen there. Great, thank you. So I think according to the program, we'll listen first to Martin Mushomba, who, as I mentioned the topic before, antibiotic resistance and staphyloxanthin from the Department of Plant and Soil Sciences at University of Pretoria. We're looking forward to listening to your talk. Thank you, Martin.
Thank you. Can you see my screen? Hello, can you see my screen? Is it visible?
Visible. You can start the talk. Thank you.
Okay, thank you. Okay, as mentioned, my name is Martin Mushomba, and I am a master's candidate under the supervision of Professor Marion Meyer. The title of my presentation is the same as the title of my master's dissertation, which is a study of induced antibiotic resistance staphyloxanthin as a prospective target against persistent pathogenic antibiotic resistant staphylococci. The layout of the presentation, I will begin by laying out the problem of antibiotic resistance, stating the aims and objectives of my study, as well as discussing the two components of this study, the comparative one and the isolation staphyloxanthin, before concluding with discussion and conclusion. So let's start by defining the problem of antibiotic resistance. By definition, antibiotic resistance is when bacteria lose their sensitivity to an antibiotic which they were previously responsive to. The various mechanisms include efflux pumps, which will pump out the antibiotic from within the bacterial cell, immunity and bypass, for which the bacteria lose the sensitivity for the antibiotic, target modification, for which the bacteria adjusts the target that was previously attached, that the antibiotic previously attached to, and inactivating enzymes which denature the structure of the antibiotic. Antibiotics came to the scene, introduced by Alexander Flemings, who is famously remembered for having left Petri dish on his desk. Might seem a very trivial kind of process, but it was actually a very deductive scientific process in which he observed that a penicillium colony, which had accidentally grown on the plate, was able to impede the growth of the staphylococci that were undergoing lysis around the structure, around the colony, whereas the rest of the colonies were growing very healthily. This was a very phenomenal discovery, one of the marvels of modern medicine. Unfortunately, over time, we have seen an increase in bacteria that are resistant to antibiotics, such as Staphylococcus aureus, various enterococci, Pseudomonas aeruginosa. And sadly, while there's been an increase in resistance, there has been a decrease in the number of companies that are researching and developing new antibiotics. In this case, plants have been seen as an important source of treatments because they are recognized as a natural source of antimicrobials, which have been found to be effective against antibiotic resistance. Plants have also been found to be particularly useful in synergistic combinations with antibiotics. An analysis was carried out, or rather a review, by Nmr Spectra and colleagues in which they looked at the four main mechanisms of resistance and were able to find various plant compounds that acted directly against these mechanisms. For example, corylagin from the pomegranate and telemagrandin or tea, which was found to work against the enzymes that denature beta-lactamase antibiotics. Another interesting one was thyme. Thyme permeability, that's allowing for an increased penetration of antibiotics. And finally, 5' recipe from the Indian snake root, which were found to impede the activity. All this information coming into context with the problem, we observe that there has been a gradual increase in diversification of antibiotic-resistant mechanisms. There has been an increase in the different antibiotics. However, as the antibiotic class up the blue arrow, we've always seen a which count produce thereby in the number of available antibiotics. Finding antibiotics just reverse the trend. Even though resistance might still occur, we will have an increase, we project to have an increase in effective antibiotics as their effectiveness is restored by these compounds. Moving to the aims and objectives of the study, the aim of my whole study was to compare the metabolites of an initially susceptible wild-type Staphylococcus with its mutated strain, and which had lost its antibiotic susceptibility. The objectives were to create an antibiotic-resistant strain from an antibiotic-susceptible one, and to compare the metabolomic profiles of the susceptible and consequently resistant strains. It was hypothesized that a wild-type Staphylococcus strain will become resistant to antibiotics after exposure to growth-inhibiting stresses, and the consequently resistant mutant will have to produce new metabolites in order to acquire antibiotic resistance. The first part of the study, which was a comparative study, required a suitable bacterium for which we selected Staphylococcus xylosis, as it was antibiotic susceptible in its wild type and was non-pathogenic. It was also similar to certain pathogenic staphylococci, such as Staphylococcus aureus and Staphylococcus epidermidis. In our first Kirby-Bauer test with the Staphylococcus wild type strain, we identified that it was susceptible to the antibiotics, as seen by the large zones of inhibition around the antibiotic discs, and this was replicated and confirmed that the wild type was indeed susceptible to the antibiotics. After exposing the same strain to starvation stress by leaving it in the same growth medium for a period of about 4 weeks, we noticed that the bacteria now became resistant and was able to grow over the antibiotic discs. The schematic in which we devised in order to study this process required first identifying that the bacteria are indeed resistant and susceptible, reculturing each of these in separate broth, and then extracting the metabolites before analyzing them under NMR analysis, which would then be used for multivariate analyses. For this part, we had we had 11 replicates. Numbers 1 to 6 were resistant strain, whereas numbers 7 to 11 were the susceptible. And what we identified was, even though there was a fair amount of similarity in all those strains, there were regions such as the part just before, started around 2.5 ppm, in which we noticed this peak here that was not visible or significant in the resistant. And inversely, we noticed that there were significant peaks that were being seen in the resistant.
I just need to interrupt you for a second. Apologies, your screen sharing seems to have stopped and we can't see your presentation anymore. Can you try and open it up again and share it again?
Please let me see my screen again.
All right, and now your image seems to have frozen. Martin, perhaps you can try just switching off your camera to conserve the bandwidth and then try and share your screen again.
Is my screen visible again?
Oh, yes, we can see your screen. It just needs to go into presentation mode and then back to you. Yeah, that was correct a little while ago.
Okay.
Okay, there you go. Thank you.
All right. Okay. So when we place the same spectra that shown in the previous slide on a PCA analysis, which is a multivariate analysis that that is supposed to compare and contrast the different compounds without having been informed of which they were, we noticed that they were able to separate between resistant and susceptible, bearing in mind that these were initially part of the same strain of bacteria. Looking at the contribution plot, we noticed that there was significant metabolites being produced in the susceptible culture, which was not being observed as significant in the resistant. And in contrast, we saw a significant portion further downfield, which were observed in the resistant but not susceptible. The second part of the study was to isolate staphyloxanthin, which is an orange carotenoid pigment produced by staphylococci to protect them against oxidative stress and UV damage. During infection, staphyloxanthin protects pathogenic staphs from host innate immune systems as it acts against the radical oxidative species produced by neutrophils and macrophages.
Sorry, Martin, we've lost your presentation again. Sorry, can you try that again? What you did before? I'm not sure what the problem is, but try and share it again.
Are the slides visible?
Fine.
Can you see the slides?
No, we've lost them again. Got it?
Are they visible now?
Martin,
Yes.
I'm sharing for you. Just tell me where you would like me to. Can you see the screen?
Yes. Next slide. Next slide. Next slide. Just next. Next. I think you're quite far away. Is it possible for you to scroll down? I'll just keep scrolling and I'll tell you when to stop. All right, stop here, stop there. Go back. So my focus on I abandoned or rather forgot 2 petri dishes on my desk on one cold Friday morning, and when I returned following Monday, next slide, I found that the susceptible on the bottom had turned orange, whereas the resistant at the top had not had the same change. Next slide. This happened after a period of 2 days in which they were left outside. Next slide. Next slide. What I determined was that the low growth temperatures of 10 degrees and lower resulted in an increase of carotenoid concentration as part of the control responses to cold shock. Next slide. This was the schematic that we came up with in order to extract the pigment. It was quite similar to that of the first study. Next slide. And as visible on the vials or the flasks on the top left corner, the first two on the left-hand side have an orange pigment, which shows that bacteria were already producing pigment whilst already in broth. And that was further elucidated on the bottom right screen, which shows that the bacteria, that's after the broth had been removed. Next slide. This was the Nmr Spectra collected for the purified extract, analyzed for the pigment. Next slide. And what we observed was specific peaks which were able to be corresponded to, next slide, to the various hydrogens, or rather the sp3 saturated carbons on the top chain for the staphyloxanthin compound. And further downfield, next slide, we observed some of the hydrogens from the sp2 hybridized regions of the alkene chains. Next slide. As visible with the yellow and the red dots. Next slide. The Lcms results, top box is from a literature source, identified staphyloxanthin at 801.2 mass per charge. And this was identified in my own Lcms result at 801 as well. And comparing this one to the resistant, next slide, the peak of the compound did not seem to appear, which seemed to confirm that the enzyme, or rather the pigment, was not present in the resistant. Next slide. Following, we had to modify this to try and enhance the signal for the staphyloxanthin. And so next slide. We decided to expand the amount of bacteria being grown by about 40. That was 40 flasks, for which we collected the bacteria separately, collected the pigment, separated it via TLC, and analyzed similar to before via NMR, UV, TLC, and GCMS. Next slide. It was already evident whilst collecting the bacteria that there was a very bright yellow rather, and this was extracted almost completely by adding methanol to the samples. Next slide. In looking at the UV absorbances, we were able to identify the absorbances underlined by the red line, which corresponded to specific chains or alkene chains that are present in staphyloxanthin. Next slide. And furthermore, the compound diprophytofluin was identified by analysis, or rather under UV spectroscopy. Next slide. Looking at the GC spectrum, we focused on the major peak at 43 minutes. Next slide. And we analyzed this by using the spectral reference library that's created by the National Institute of Standards and Technology. Next slide. Opening the window to the NIST program, we first identified squalene, which corresponded to the peak previously seen in the GC slide, and that was at the probability of 59%. Next slide. And with the blue spectrum at the bottom being that of squalene, the top spectrum being that of the compound we were analyzing. Next slide. When we compare with the staphyloxanthin, we found the same 24 carbon chain, alkene chain that is present in staphyloxanthin, with the difference of less hydrogens being less saturated regions. Next slide.
Significant peak of 26 minutes. Next slide.
We identified a tetradecanoic acid chain which corresponds to, next slide, the saturated sp3 chain that is present in cephaloxanthin, at the top of cephaloxanthin, with 14 carbons and the carbonyl region. Next slide will be the discussion and conclusion. Next. The 12-methyl tetradecanoate and 4,4' diphytorine chains were detected by UV spec, TLC, proton NMR, and GCMS analysis. The 4,4' diponeurosporine, which was the bright yellow seen on the biochemical pathway on the right, was not detected, but this may have been a result of reaction of the compound with light, which would have resulted in saturation of the compound, or changes in pH, which would have resulted in reduction or addition of hydrogens. Next slide.
The difference in strains was noted
by Marshall and Wilmoth, and it's possible that cephaloxanthin was produced, albeit in small quantities. Next slide. Cephaloxanthin, as mentioned before, is a secondary metabolite used in virulence mechanisms, neutralizing radical oxidative species produced by macrophages and neutrophils. Thus, next slide. Focusing on inhibiting virulence mechanisms instead of essential functions means bacteria will not experience the stresses, as we've seen in my experiment, that forced them to adapt and become antibiotic resistant. Next slide. One example of these was the study carried out by Lee and colleagues, who found that about 12 different plant flavonoids were tested against the cephaloxanthin-producing bacteria and were found to reduce production of the pigment without inhibiting staphylococcal growth. Next slide. In conclusion, an antibiotic-resistant mutant of Staphylococcus was obtained. Cephaloxanthin was partially isolated from the wild type, and the difference between wild type susceptible and resistant mutants were corresponded with changes in metabolite production. Next slide. What I would project thus is a lot of antibiotic resistance is focused on antimicrobials that limit bacterial growth. More work on virulent mechanisms that are not essential would increase the effectiveness of antibiotics while not allowing bacteria to become resistant. Such targets as cephaloxanthin. Next slide. I'd like to acknowledge my supervisor, Prof. Mario Nmr, for his assistance, as well as Dr. Emmanuel Chikalange, my good friend Sievas, and Mama Lucy at the chemistry department for their help in LCMS and GCMS analyses. Thank you very much.
Thank you so much, Martin. That looked like a fascinating project, very lucidly presented. Some very important issues about what we should be looking at in terms of antimicrobial resistance and where to target these bacteria. I don't see any questions in the chat, but we can keep an eye on the Q&A and the chat as we're running a little bit short of time. So I'd just like to see if Chika is in the presentation room. Thank you very much, Martin. Fantastic presentation.
I'm very sorry, Lindy. I think there's a question.
Oh, is there a question?
I can respond to it. We can, so it's not so long for the sake of time. Now respond on the chats.
Okay, that's perfect. Okay, thank you so much. Appreciate that.
Thank you.
Thank you very much, Chika. I see you in the room there. It's my pleasure to present one of our PhD students, Chika Ogwadike, who's in the phytomedicine group at the University of Pretoria. And she's going to be talking about some of her preliminary PhD research on Celestraceae, 2 Celestraceae, and 2 alien invasive Cratulaceae species with potential against mastitis and their different bioactivities. Thank you, Chika. You can go ahead and share your slides.
Thank you, Prof. Can you see my slide now?
Slide, so you can go ahead with your presentation.
Okay. So thank you for introducing the presentation also. And this is the outline of my presentation. We are working on finding solution to the disease called mastitis. And mastitis is the inflammation of the mammary parenchyma or breast. It affects cattle and humans and is caused by infection of microorganisms like bacteria, fungi, and in seldom cases, viruses and algae. Bacteria are major causative microorganisms. Mastitis can be subclinical or clinical. When it is subclinical. It is not visible, but can be confirmed by somatic cell count, together with presence of microorganisms in milk. Whereas when it is clinical, inflammation can be visible and can be accompanied with ulceration and abscesses. Inflammation, a major feature of mastitis, under normal circumstances, promotes healing, but sometimes it can become uncontrolled and go on and on and on. And when this happens, it can result in cell damage as well as oxidation. And oxidation equally is harmful to cells and surrounding tissues. The major causative organism of mastitis is Staphylococcus aureus. And this Staphylococcus aureus has developed resistance due to the indiscriminate use of antibiotics. In 1960, methicillin was developed to be used against Staphylococcus aureus. In the following year, methicillin-resistant Staphylococcus aureus was isolated. Over the years, it has been found that methicillin-resistant Staphylococcus aureus is also resistant to other classes of antibiotics, and synthetic antibiotics are not only failing in activity, they also have harmful side effects. Resistant Staphylococcus aureus is prevalent in South Africa, as well as all over the world. Mastitis has economic effects and consequences. In affected cow, it can lead to decrease in milk synthesis, as well as low quality of milk. This is an example of bad milk from affected cow. Mastitis also leads to huge financial losses to dairy industries around the world. Plants can serve as alternative sources for finding anti-infective agents. And this has been proven in ethnomedicine and chemotaxonomy. In ethnomedicine, traditional medicine has been used for ages to treat microbial infections. And in chemotaxonomy, through research, it has been found that certain plant families and the plant orders have antimicrobial activity, and these are being documented in databases. Also, through research, it has been found that plants act by either reducing the growth of microorganisms or killing them, and also by inhibiting the entry of microorganisms into cells, as well as by reducing the effects accompanied with synthetic antibiotics. So the aim of our research is to find bioactive extracts from selected medicinal plants that have the possibility of preventing and/or treating mastitis. The microorganisms we use in this research are Staphylococcus aureus ATCC strain and 6 mastitis clinical isolates of Staphylococcus aureus, which we got from the milk laboratory of University of Pretoria. For plants, we first screened 38 plant extracts belonging to 16 plant species for antibacterial activity. And 4 most active plants were selected. 2 of these 4 most active plants belong to the family Celestraceae, and the other 2 belong to the family Crucilaceae, and they are invasive plants in South Africa. These are the plants. Muracinia frangula and Methenus odonta belong to the Celestraceae family, whereas Kalanchoe pinnata and Bryophyllum pinnatum belong to the Crucilaceae family. Plants we are choosing from the Celestraceae family following the research did by Paul and Elof, in which they found that the Celestraceae family has potential for antimicrobial activity. Plants were also chosen from Crucilaceae family because of their use in traditional medicine. Our plant material were extracted with acetone and ethanol. We want to extract all active compounds, irrespective of their polarity. So we chose these solvents. Ethanol was to extract polar compounds, whereas acetone, being a wide spectrum extractant, was to extract intermediate polar compounds as well as non-polar compounds. We did antibiogram assay using Edgar-Dix diffusion method, and it was done to ascertain the antibiotic susceptibility of the organisms we use in this research. These 10 antibiotics belonging to these 7 classes of antibiotics were used for this assay. We also checked the antibacterial activity of our plant extracts, first on the ATCC strain, and then followed by testing them on the mastitis clinical isolates. We further investigated the cytotoxicity of our plant extracts against Vero cells and bovine demis cells and determined the selective index values. Further, our plant extracts were assayed for antioxidant activity, anti-inflammatory activity, as well as their effect on cell viability. Data obtained were analyzed with ANOVA and Excel. Results and discussion. Our antibiogram assay revealed the antibiotic resistant profile of the organisms used in this research. These are the organisms used. This is Staphylococcus aureus ATCC strain from STA1 to STA6, or the mastitis clinical isolates. With the inclusion of the intermediate resistance, it was observed that all the clinical isolates were resistant to at least 3 antibiotics. 4 of the clinical isolates were resistant to 4 antibiotics, whereas the ATCC strain was only resistant to 2 antibiotics. The antibiotics that these organisms were resistant to fell into 3 classes of antibiotics, which are the penicillin class, the polymyxins, and the aminoglycosides. M-frangular acetone extract had the best mean antibacterial activity on all the 7 bacterial strains tested, followed by Carpinata extracts. These are the results of the cytotoxicity assay we did on Vero cells and bovine demis cells, and also the average selective index values we got. Selective index values greater than or equal to 1 indicate more activity on bacteria than cytotoxicity to mammalian cells. M-frangular, Carpinata, and Bipinatum extracts have selective index values equal to or above 1. The higher the selective index value, the better. For antioxidant assays with DPPH and ABTS, Carpinata extracts had the best activity, followed by diphylum ethanol extract. And the activities of the Carpinata extracts were comparable to the activities of the positive controls, Trolox and ascorbic acid. Also, for anti-inflammatory assays, checking for the inhibition of lipoxygenase. Again, Kalanchoe pinnata extracts had the best activity, and their activities were comparable, again, to the activity of the positive control, quercetin. Anti-inflammatory assays checking for the inhibition of nitric oxide, And this assay, at the highest concentration, we found that Bryophyllum pinnatum extracts had the best activity, with nitric oxide inhibition above 78% at the highest concentration. They were followed by Carpinnata extracts. Activities observed with M. odonta acetone extract and M. frangula extracts were as a result of toxicity. As you can see, at the highest concentration, their activities were more than 100%, as you can see here, indicating that it was no longer nitric oxide they were inhibiting. This is also confirmed in the next slide. Quercetin was the positive control used here. This is the result of the effect of our plant extracts on cell viability using activated raw macrophages. All Bryophyllum pinnatum and Kalanchoe pinnata extracts have cell viability above 90% at all concentrations, indicating their safety. M. frangula extracts showed the lowest cell viability at the highest concentration, indicating toxicity, and also confirming that the activities observed with them in the nitric oxide inhibition assay were as a result of toxicity. Here, you can even see that at the highest concentration, the cell viability was down to 5%. Conclusions. Carpinnata and B. pinnatum extracts showed good bioactivity with low cytotoxicity and are therefore potentially promising for the management of mastitis. All the mastitis clinical isolates of S. aureus showed resistance to at least 3 antibiotics. Further work is ongoing on active compounds isolation. I thank my supervisor, Professor Lindy McGow, and my co-supervisor, Professor Pitzer, for their different kinds of support. I also thank all my colleagues in the phytomedicine program. And I as well thank University of Pretoria for funding this research. My thanks also go to the organizers of this conference for giving me the opportunity to present our research. These are some of the references used in this presentation. Thank you all for being here and for listening to my presentation.
Thank you very much, Chika. I must say I'm looking forward to the results of further studies with formulation and potential in vivo studies, as it's such an under-investigated field. Thank you. Are there any questions for Chika from the audience? Just waiting to see if something pops up on the screen. Oh, something here from Service. Thank you for the question and thank you for the interesting talk, Chika. He says, in what did you redissolve your extracts for the assays? And did you investigate using an antibiotic along with the extracts?
Hello, Prof.
Oh, hi. Okay, did you hear the question? He's asking, what did you redissolve your extracts in for the assays? And what was your antibiotic positive control?
Okay. For the antibacterial assay, I redissolved my samples with acetone.
Okay, and the positive control you used?
Gentamicin.
Okay, that's great. Thank you. Any other questions? Okay, very interesting results, and hopefully we'll come up with some lovely formulation to be developed into a product at some stage. I would like to thank very much the presenters in the session. Are there any other questions for any of the presenters that might have come up while you were listening to the other talks? Okay, I don't see anything popping up there, but thank you for keeping largely to the time and for some fascinating results that we've seen of this research. And we're looking forward to looking at the next stage of what comes next of these interesting postgraduate studies. So congratulations to all. I'd like to wrap up this session, thanking the participants as well for their questions and for their participation. Thank you, everyone. Good night.
Thank you,
and see you tomorrow. Thank you.
Prof. Lindy, for the last session of the day. I would like to take this opportunity now to thank all the presenters, starting with our plenary speaker, Dr. Matt Bias, and all the other parallel session presenters who kept us busy for the whole day. The talks were very interesting and very educative, so we had a good day for our conference. I also need to thank the audience. I think you keep on giving us good contributions through your questioning and also comments, which is fine. I would like now to specifically thank all the parallel session chairs for the 3 sessions that we're having, Dr. Aidama Duna and Prof. Sandra Bernard, Prof. Salome Mahlo, Prof. Jackie Barna, Prof. Lindy Mago, and Dr. Mariette Jackson. Thank you very much for chairing the sessions. You kept us busy and it was exciting. I also would like to thank the technical team. I think today we're having a lot of hiccups with the internet connections, but somehow you managed to keep us on track, which is quite good. And it's actually encouraging when we are going towards the end of our conference that we can at least keep on tracking our time and managing to bring our presenters in or jostling around the presenters. If we miss one, we can bring the other one and then exchange them. It was quite well handled that I think we didn't experience much challenges. At the same time, I think I need to thank everyone for being here for the whole day. I think it's also exciting that we are remaining here as a good team. I have one announcement before we close. Yesterday, I indicated that the photo competition was reactivated. So what we need to do is tomorrow morning, starting from tomorrow morning, we need delegates to start voting for the best photo. So I will remind you again in the morning about it. But what will you do? What will you will do is to go to the website. Then you click on photo competition. After that, you go on vote. Then you vote for your best photo there. So we'll open that session from the time that we start tomorrow at 9, then close at 1. So all those want to participate in choosing the best photo will do that tomorrow. So we are now closing for the day, and we'll meet tomorrow for our last day of, which is the final day for the conference. So I wish you a restful evening and a good night. We'll meet again at 9 o'clock tomorrow. Thank you very much, and goodbye.